Hello everyone, my name is Esteban. I am really happy to be this morning with all of you at the CFGA WFGA 2021. I am a research associate working with Bart Thomas at the University of Alberta. Also, this presentation is going to be about a recent paper that we published with Jan Busquet from the Laval University. It is called SNP-based analysis reveals unexpected features of genetic diversity, parental contributions, and pollen contamination in a white spruce breeding program. So first of all, we are always looking for the most accurate ways to monitor genetic diversity, parental contributions, and pollen contamination, particular for tree improvement programs. So SNPs are so far the most accurate molecular markers for that, and we can track the diversity and pollen contamination for the different populations, such as natural stands, breeding population, progeny trials, and particularly seed orchards and seed orchard seedlots. That is to ensure that we have enough diversity to accomplish like normativity, and also to get the industry have access to the best gains through backward and forward selections, and at the end to have a balance between diversity and gain. And pollen contamination, it is one of the most crucial topics to talk about the seed orchards. And it is because it is a potential problem that affects genetic gain and also needs to be completely accurate. So this is an example of, let's say, wild stand that can be very close to the orchard, and both are from the same species. So if we imagine, for example, a 50% of pollen contamination, it can cause a loss of half of the genetic gain. So that's why we focus our attention for this experiment in a program called G1. As you can see here, around all the province, we have different white spruce program. So what we did with the G1, we went into the founders, into the seed orchard, and we collected different seedlots and progeny trials in different places around the province. How many? We collected 151 individuals in the founders, 5 seedlots with a total of 525 trees or seedlings. And in the progeny trials, we collected 33 trees from each family. What we wanted to accomplish, measurements of genetic diversity through effective population size parameter, to estimate male and female contributions in the seedlots, and pollen contamination levels also on the different seedlots. So as methods, we went into the progeny trials and founders, and also we grew the seedlings of the different seedlots. We collected the needles, we stored them in cold and then into the freezers. We extracted the DNA. The DNA was placed into different into the SNP chip. In the different cells, the SNP chip were standardized by the Jan Busquet lab at the Laval University. After a couple of weeks, we obtained all the SNPs from each individual. We cleaned the SNPs using the Genome Studio. We used Genolex to obtain the relatedness and diversity parameters, and Servos to get the parental assignment. Finally, with the different relatedness coefficients, as 0, for example, for unrelated trees, as 0.25 for half sieves and 0.5 for full sieves, we could get all the pedigree reconstruction. So what we found? First, for the diversity and contributions, in the y-axis, I am showing the effective population size, and in the x-axis, the different populations. For founders, we had 500 as population size, and it decreased with the seedlots from 2003 and 2009, as well as we found an increase with the relatedness from founders to the different seedlots. But after eliminating 65% of the trees in the orchard, which is 2/3, we found a huge and significant decrease from 96 to 12, which is 5-fold, in effective population size, which is diversity, as well as a huge increase in the relatedness, which is 8-fold, from 5 to 40. Also, for contributions, we have in here the participation of females and males for each seedlot. Before rogging, we had equal contributions for females and males, but after rogging, we found a significant change and an equal contribution of mothers and fathers. Let's talk about pollen contamination. We found in here in the x-axis, I am showing the different pollen contamination levels that can go from 5 to 50% for the different years. And we wanted to see if there is a relationship or a correlation between the wind direction and the pollen contamination. And we have found 92% of correlation with an exponential trend. Then we were wondering if we had like a source in the site, and we found definitely a contamination source that can increase the external pollen depending on the amount of wind that we have from this white spruce orchard to the target orchard of a study. Then we analyzed the pedigree from the progeny trials, and we found that in the colored dots, all the mothers or females, and in the black dots, the males. What is interesting here is that just 11 males were mainly contributing for the whole pollination for the different progeny trials, and unfortunately, we found 30% of pollen contamination for the whole progeny trial, which is external pollen. As conclusions, so recommended distances of 1 kilometer that we usually find in the literature are insufficient to ensure low contamination levels in seed orchards. Second, random mating does not happen in operational tree breeding, as we all know. So a careful design of breeding program is definitely necessary. Third, tree breeders should avoid severe roggings to prevent increases in relatedness and genetic diversity losses. And fourth, we recommend the adoption of genomic tools and the use of SNPs to advance the tree breeding programs here in Alberta. So as perspective, we definitely are suggesting genotyping for accurate pollen contamination assessment, progeny testing of selection of parents, as well as estimates of diversity with effective population size calculations, and knowing the male-female contribution in seedlots. The first two to increase gain, and the last two to measure diversity for the industry and the government, respectively, and to have a balance between gain and diversity. I would like to thank our lab sponsors, which are in the bottom of this slide. Also, all the team that has been very helpful all these years for all the work that we have at the Thomas Lab, and including the different lab coordinators, the postdocs, the technicians, and I would like to thank, of course, Barb Thomas and Jean Busquet for all the knowledge that we have all put on this paper. And finally, I would like to thank the CFGA and WFGA for this opportunity to present our paper. So I am open to questions. Thank you very much.
Hello everyone, my name is Dawei Luo and I'm from University of Alberta. I'm a PhD candidate from Thomas Tree Improvement Lab. Today I'm very happy to make a presentation of one of my research. Before I start, I'd like to give a thank you to co-authors of this research. Dr. Greg O'Neill, Dr. Yu Qingyang, Dr. Esteban Galeano, and my supervisor, Dr. Barb Thomas. And also a thank you to all sponsors for their financial support. As we know that climate change has been a big challenge in past decades and has been observed with significant impact on forestry. In Alberta, an increase of mortality, growth reduction, and the shrinking forest land base associated with climate change have all been reported, and these trends are projected to continue in the near future. The continuous climate change stress is going to have impact on two major commercial tree species in Alberta, white spruce and lodgepole pine, and they account for about 90% of annual reforestation. In 2019, 65% of white spruce seeds and 21% of lodgepole pine seeds were accounted for by genetically selected seeds. An increase of investment on tree improvement has made it necessary and challengeable work to figure out how climate change will impact improved trees, especially these two species. Therefore, the main job of this study is to incorporate both climate change and genetic gain into growth and yield. To answer this question, we made use of data from both progeny and provenance trials. This map represents all provenances and tested sites for white spruce and lodgepole pine that's used in this study. The orange dots represent the provenance and the cyan dots represent the tested sites. We have over 500 white spruce families and over 1,000 lodgepole pine families selected from the progeny trials, 50 white spruce populations and 44 lodgepole pine populations selected from provenance trials. This figure shows the range of mean annual temperature and mean annual precipitation for progeny and provenance trials for white spruce and lodgepole pine. Except the lodgepole pine provenance trial, we have a wide range of temperature and precipitation for the climate origin and the climate of tested sites for white spruce and lodgepole pine. Making use of above data, we first use provenance climate and the climate transfer distance to estimate the height proportion of each population tested in different sites relative to their average height at the provenance. Hereafter, this value is denoted as height p-value. Then we build the adjusted pool transfer function to estimate height p-value, and we merged the height p-value with genetic gain and use them to modify the top height and the site index. The growth and yield model used in this study is called the Growth and Yield Projection System, or GYPSY. And in GYPSY, site index and the top height are the main drivers for the stand yield. The corresponding stand yield could then be predicted according to the modified top height curves. We selected 3 climate change scenarios in this study, including low scenario, intermediate scenario, and high scenario, and they represent different levels of greenhouse gas emissions and the consequential global warming level. After comparing available climate variables and different functions, we selected a second-order polynomial function to predict the population height p-value response to climate. And we found that the mean coldest month temperature is strongly correlated with the height growth of white spruce, and the mean annual precipitation is strongly correlated with the height growth of lodgepole pine. And this figure here shows the parameter estimates for the final adjusted pool transfer functions for white spruce and lodgepole pine. And in our results, we have found that the height p-value was independent of age and therefore was excluded from the final functions. The height p-value variation, as expected, shows single peak features, as shown in these heat maps. For white spruce unimproved and improved seed lots, their optimum MCMT is around -16 degrees. For a population from a colder place, the increase of temperature will benefit their growth. And for lodgepole pine, their optimum mean annual precipitation is about 500 millimeters. And for the population from a drier place, the increase of precipitation will have positive effect on their height growth as shown by the green color. And on the other side, for white spruce, if the provenance MCMT is higher than -16, the increase of future climates will have negative effects as shown by the red color, and the decrease of temperature will have a positive effect on their height growth as shown by the green color, and the same for the lodgepole pine population. If we merge the height p-value into the top height curves, we will have something look like this. Different color of the curves represent the different climate change scenarios. And now we only focus on the black one and the red one, which is no climate change scenario, which is the black one, and the red one is for the high scenario of climate change. The dashed curve represents the improved seed lots with 10% genetic worth, and the solid line indicates the unimproved seed lot. In the hottest sites, we can see that the increase of temperature in the future will have significant negative effects on the height growth, with almost over 50% height growth off. And in the coldest sites, we can see that the increase of temperature will benefit the tree growth, and we can see the top height is almost doubled for this site. And for lodgepole pine, unlike white spruce, in all selected test sites, we found that their height growths are going to decrease regardless of climate change scenarios, and we can see a significant drop of top heights with aging, which indicates the potential mortality for the top height trees in the high scenarios. Applying the adjusted pool transfer functions to all provenances and tested sites, we will have a map look like this. For white spruce, in the northern Alberta, they are going to have an increase in height growth. And in the middle to southwest Alberta, the increase of temperature in the future will have negative effects on their growth for both unimproved and improved seed lots. They will show the similar pattern in the future. And for lodgepole pine, in most of those sites, the color is orangey and red, which indicates that they are going to have a decrease in their height growth, except some sites in the north, which is shown by the green color. If we compare the performance of improved and unimproved seed lots, we will find that in the locations where the climate change is going to benefit the tree growth, the unimproved seed lots are going to benefit more, which is in the northern Alberta. And in the locations where the climate change is going to have a negative effect on the tree growth, the improved seed lots will remain their growth superiority over the unimproved. In conclusion, future climate change is expected to increase white spruce growth for those populations originated from cold provenance, but to decrease growth for populations from warm provenance. For lodgepole pine, the growth decrease is provincial-wide, except some sites in the north. For both species, the unimproved seed lots will benefit more than the improved seed lots in locations where climate change will benefit tree growth, but improved seed lots will alleviate the negative effects of climate change in where stand yield is going to decrease in the future. And that's all for my presentation. Thank you all for listening, and all questions are welcomed.
Hi everyone, my name is Yue, and I'm a master's student with Dr. Tony Wang at UBC Forestry. Today I'm presenting my master's research on using landscape genomics to delineate lodgepole pine seed and breeding zones. Well-defined seed and breeding zones are important for developing adaptive forest resource management strategies. And traditionally, seed and breeding zones are developed based on the relationship between adaptive traits and climate variables through a series of provenance trials and common garden experiments. but collecting phenotypic data is expensive and time-consuming and are only available for certain species. Here, we offer an alternative by looking at genomics data and use landscape genomics approach to find a relationship between genomic variation and climate variables, specifically through the use of the gradient forest model. My study focuses on lodgepole pine, one of the most ecologically and economically important tree species that's widely distributed in Western North America. It occupies a large climate gradient, which made the species important to develop effective seed and breeding zones. The Illingworth Provenance Trail and the Adapt Tree Project have both accumulated sufficient phenotypic and genetic data for lodgepole pine. The objectives of this research is by building a gradient forest model to identify major climate variables driving lodgepole pine's local adaptation, to delineate seed and breeding zones for lodgepole pine, and to predict the spatial distribution of the genetic offsets for lodgepole pine under future climate. In order to build a gradient forest model, we used 36,384 SNPs collected by the Adapt Tree Project and 20 annual climate variables generated by Climate.NA. The predictor importance graph we see here is one of the GF model's outputs. We can see all 20 climate variables used to build the model have been ranked by their importance, meaning how important it is to drive the lodgepole pine's local adaptation. And we could see that the winter temperature-related variables and the moisture-related variables are the major drivers. Another important output of the GF model is the cumulative importance graph. These nonlinear relationships provides us information of how the GF model can transform environmental gradients into genomic gradients, which serves as the basis for the GF model prediction process. When predicting, we input only climate data for the coordinates we wish to look at, and the GF model will be able to transform these data into genomic information that's multidimensional. To visualize this, we used principal component analysis to see the first three PCs. To take a closer look at this graph, to our right here is the spatial distribution of lodgepole pine's genomic variation. To note that similar color on this map indicates similar genomic composition. But the points presented here are continuous, and they need to be clustered to achieve the objective of the study, which is to delineate seed and breeding zones. The question comes to how many zones should we cluster these continuous points into to get a relatively reasonable seed and breeding zone? We have tested numbers of clusters from 2 to 16. The optimal number of clusters was determined by a significant reduction of the within cluster variation when an additional cluster was added, which indicates a decreasing benefit of adding more clusters. Here, we were able to estimate 8 clusters for our breeding zone purpose. And this is the 8 breeding zone predicted by the GF model covering the distribution range of lodgepole pine in BC and Alberta. But now we need to validate if this genomic-based breeding zone is effective and reasonable. A valid comparison would be with the breeding zones by Lippi et al. Their delineation was based on phenotypic data, specifically seedling adaptive traits for the same population used in our research. We could observe an overall similar pattern, and for regions that did not line up, it is probably due to lack of samples. In this comparison, an advantage of our genomic-based breeding zones is that we have continuous zone distribution across the provincial borders, whereas Lippi zones are more discrete. This suggests that the GF model can provide an alternative for delineating seed and breeding zones. We also delineated 4 seed and breeding zones for the GF model for lodgepole with the GF model for lodgepole pine in BC. This is to facilitate a better comparison by matching the number of zones proposed by Ukrainians et al. We found that they were roughly similar, and where there was poor alignment, the GF-based zones appear to be more reasonable. For example, the GF-based zones separated the coastal regions from the interior and were better aligned with the BEC zones for the province. So back to the GF-based 8 breeding zones with color adjusted to match the BEC zones. We could observe a similar pattern when comparing to the BEC zone map to our right here. This is because climate gradients driving landscape genomic variation in lodgepole pine could also shape the terrestrial ecosystem difference in the province, and the GF model was able to reflect such differences. At last, we look at the projected genetic offsets for lodgepole pine in BC and Alberta in 2050s under scenario RCP 4.5 and 8.5. Our projection here assumes populations are adapted to the current climate. Color from blue to red represents low to high genetic offset for lodgepole pine. And as expected, a more severe climate change scenario projects higher genetic offsets across a larger range. This highlights the vulnerable regions if we continue to use local seeds under the changing climate. And our projected results could also potentially be used as a reference for assisted migration. Here are the conclusions for this research that we have covered. And the main takeaway is that the GF model can be used as an effective alternative approach to delineate seed and breeding zones using genomics data. I would like to thank the ADAPT Tree project for generating the lodgepole pine genomic data used in this research. And I would also like to thank valuable comments and suggestions from lab mates and funding from CSC and INSERC. Here are the references. Please let me know if you have any questions and suggestions. Thank you.